This is another review video, and we're going to tackle Unit 3. We're going to run for about 20 minutes, and then we'll just stop wherever we're at. In the third 9 weeks, we did bacteria, and then we did protist, and then we did fungi. So we're going to go back and start with bacteria here. We started off, we talked about the shapes. We said, I don't want to change this real quick. Give me just a second. Let's change this to blue. So we said we have three shapes. We have rod on the left. We have sphere, which is known as cocci, and then we have spirochetes. So, whoops. So three different shapes. And then we went into the first part of this as we tackled the anatomy or the structure of prokaryotic cells. So bacteria, and we're going to talk later, have two different domains, archaea and eubacteria. But in terms of bacteria, they're all prokaryotes, and they have similarities. So all cells have cytoplasm, they have ribosomes, and they have a plasma membrane. So it doesn't matter if you're eukaryotic or prokaryotic. Those are the three things that you have. Some of the differences we talked about is prokaryotic ribosomes are smaller. And then we talked about the things that are common to prokaryotes. So the ribosome, cytoplasma, and plasma membrane are all cells no matter what. Prokaryotes also have a cell wall, and they have this circular DNA. So those are characteristics of prokaryotes. So if we look at, let's start with the circular DNA, and it's found in a region called the nucleoid. Remember, prokaryotes don't have membrane-bound organelles, so they don't have a membrane-bound nucleus, but they have a region where the circular DNA is formed, and it doesn't look circular, but if you unwound it, you would see that it is in fact a circle. In addition to this circular DNA, they have what are called plasmids. Plasmids are smaller ring-shaped pieces of genetic material, and those can be exchanged between bacteria. In addition, bacteria have a cell wall, and we talked about gram-positive and gram-negative. And that's a little bit in a few slides. We'll talk about that more. So we've got different types of cell walls. Capsule is something that may or may not be present. Capsule it's sticky, and it allows the bacteria to adhere to surfaces. And again, not all bacteria have capsules. These pili, there are different projections out of bacteria. We have fimbriae, we have pili, we have flagellum. We're going to talk about those specific. I'm going to save that for later. And that is about it, I believe. Yeah, we'll talk about pili here in a bit. So I'm going to leave that off of this screen. All right, gram-positive versus gram-negative. So these are those cell walls we were talking about. And we did a lab where we stained bacteria to determine if they were gram-positive and gram-negative. So here you have the gram-positive profile. Here you have the gram-negative. And what we see is we have our plasma membrane. So this is the cyto, I think that's a typo, where we've got the phospholipids, and we talked about phospholipids with their hydrophilic and hydrophobic. But then the cell wall for gram positive here, cell wall for gram negative is here. Whoops. Yeah. And what we see is that for the gram positive, you have this very thick layer of peptidoglycan. And when you put that into crystal violet, it turns purple. So you can tell that you have gram positive by that. For the gram negative, you get a very thin, this is the peptidoglycan again, but it's in between two plasma membranes. So I've got an inner plasma membrane and I've got an outer plasma membrane. And on top of that outer plasma membrane, we have lipopolysaccharides. And what we're going to see is those are toxic when that breaks down. But because of this outer plasmic membrane and the thin peptidoglycan on the inside, when you put gram negative into crystal violet, they do not turn purple. So they're negative for that color. Okay. moving on. We talked about osmosis, bacteria with osmosis, and you have got hypotonic, isotonic, hypertonic. If you're in a hypertonic solution, there is more solute outside than there is inside, and so the water leaves. If you're isotonic, it's not that you don't have water movement, you do, but the water in and the water out equal each other, so it's at equilibrium. And if you're a hypotonic solution, you have less solute outside the cell so that water enters in. We talked about some hypertonic solutions. We gave the example of honey. So people talk about the fact that honey is good for resisting bacterial growth. And the reason is it's a hypertonic solution and things cannot grow in it without shriveling. And we talked about swelling, lysing, shriveling. So again, we spent quite a bit of time on this. Make sure you go back, and if you don't remember it, look over these terms. Okay, additional things with the cell surface. So this is an example of a capsule. And number two, if that capsule is very disorganized, we refer to it as a slime layer. And then in the third one, we have a biofilm. And we talked about biofilms in great detail. So we basically said biofilms are when you get more than one type of bacteria, you get different species, and they exist together cooperatively, and they can send out signals to recruit other organisms. And once they reach a certain point, they grow to a certain size, they will split and release bacteria to form a new biofilm. But the problem is that a lot of times these are, are we doing biofilms later? I don't remember. Biofilms can be difficult to kill, and so they result in a lot of infections and problems with medical equipment. I think we have it in our notes later. I'm going to wait for that. The other thing we talked about, so here we have fimbriae. Here's the flagellum, and that's a sex pili or a pilus, are often used to exchange plasmids. The fimbriae are used to adhere to surfaces and adhere to each other. So those are for adhesion, and the flagellum obviously is for movement. Also, in addition, this capsule provides protection, the capsule and the slime layers. And the difference here is this is organized, this is pretty disorganized. Bacteria have the ability to form what are called endospores. So if they get into a condition where there's not a lot of nutrients or they're in a hostile environment, they will, inside of the cell membrane, cell wall, they'll begin to take this piece of genetic material here. So this is genetic material. And it's called an endospore. Endo is inside, and spore is a structure that's got a hard, we're going to talk about spores and fungi, but it starts to form this coat. And we end up with an inner membrane, a cortex, an outer membrane, and a coat. So this is a very, very thick, very protective structure around this genetic material. As the parent, they call it the mother cell, the parental cell dies off, this remains. And what happens is that at a later date, if this goes into conditions where the environment is not hostile, it can reform into the original bacterium. So it's a survival tactic, and they have found endospores in all kinds of locations. Next, we have got flagellum, and we said that a prokaryotic flagellum and a eukaryotic flagellum are not the same thing. And in fact, later we're going to talk about the difference between archaea flagellum and eubacteria flagellum. But overall, even though they're the same size on this diagram, prokaryotic flagellum are smaller, eukaryotic are larger. This rotates. The prokaryotic rotates. The eukaryotic moves back and forth. That's what this is showing back and forth motion versus rotating. This has flagellin protein. This is tubulin. And the prokaryotic flagellum exists outside of the cell membrane. And what we see with the eukaryotic flagella is that cell membrane wraps all the way around. In Archaea, their protein, yeah, so Archaea has a different protein. So we said this was flagellin, this was tubulin, and Archaea bacteria have archelin. That's their protein. Another thing we talked about was the genetic material. So we said already that prokaryotes have a circular chromosome. Eukaryotes have a linear chromosome. When we get ready to copy these chromosomes, the prokaryotic chromosome opens at one replication point. So this is called a replication fork here. And over here with the eukaryotic, they're showing multiple points of replication. There's so much genetic material in eukaryotes that if you only had one replication fork, it would take forever to copy. So eukaryotic genetic material has to have multiple replication. So if we go down the checklist, prokaryotes are circular, eukaryotes are linear. No associated proteins. So over here, I see this is a histone, and this is the chromatin, the loose genetic material of a eukaryotic cell. When we condense into chromosomes, there's a lot of chromatin in a eukaryotic cell. And in order to get it to condense, it wraps around these proteins. Prokaryotes don't have that. Prokaryotes have plasmids, eukaryotes do not. Prokaryotes have one chromosome, eukaryotes have two or more. In addition, eukaryotic has something that we talked about called introns and exons. We added that to the list, and we talked about what introns and exons were. We then compared the reproduction of prokaryotes and eukaryotes. So if we have to create new cells because we're growing or because we were injured, eukaryotic, we undergo mitosis. We have a prophase where they condense, and this is actually a diploid cell before, I'm sorry. Actually, this starts with metaphase where they line up, anaphase where they pull apart. Prophase isn't being shown here. So if it was, they'd be duplicated if they were in prophase. Binary fission with prokaryotes. I'm going too fast now. I'm starting to make mistakes. And there's other organisms that undergo binary fission. But we're talking in terms of prokaryotes and bacteria. So again, you get one replication fork, and that divides, it moves to opposite ends of the cells. This pinches in, and we get two daughter cells. And for both of these, we get two identical daughter cells. And we're going to talk about, well, where does the variation come in? We're going to talk about that later. Now, you can have mutations in both of these processes, but prokaryotes, their populations are usually huge, and they have a shorter lifespan, which means we have generation after generation very quickly. That makes mutation a much bigger factor in terms of time frame and in terms of, as compared to our lifespan, in terms of how quickly it happens. So if our lifespans are the templates, we look at prokaryotes as mutating fairly frequently. But you've got to have variation, right? That natural selection has to have variation for it to occur. We're not going to go back over this, but we went back through meiosis in eukaryotic cells, and we said that we get the crossing over. So right here where we get crossing over, and it shuffles up the alleles. In prokaryotes, there are three ways that we can get variation. We can get transformation, we have conjugation, or transduction. Transformation is getting it from the outside. Conjugation is swapping bacteria to bacteria. Transduction is when a virus inadvertently takes genetic material from one bacteria and copies it to another, specifically called a bacteriophage. So transformation, we take it in, this is the environment, and you can take it in plasmid form, or you can take it in a fragment form. For it to be successful, it has to be, if it's a fragment, it has to be incorporated into that large circular chromosome. If it's not, it'll just be broken down. Plasmid doesn't have to have that. A plasmid can exist just as it is. So plasmid transformation and fragment transformation are not exactly the same thing. Transduction. So here's that bacteriophage. The red is the viral genetic material. The blue is the bacterial genetic material. So during infection, the virus takes over the cell, causes it to make the components of the virus that get assembled. And here you can see the little piece of the initial host's genetic material got incorporated. So what happens is when that virus then goes on to infect the next bacterium, it injects that piece of genes and it gets incorporated into the second bacteria. And you had down here a key. So that is transduction. Conjugation, that is bacteria to bacteria. Again, we have a couple of different things. So you have to be F positive. You have to have an F plasmid to be a donor, and you are F negative as a recipient. And once you transfer, this occurs through a pilus connection known as a mating bridge. And that is primarily what the pili are used for, is conjugation. That gets sent through the pilus, through the mating bridge, and it gets sent to the second bacterium, and now they're both F positive. Another possibility, oh, and we said that if you are, you could have R positive, R negative, and that's antibiotic resistant plasmids. So we kind of classify those as their own thing. Here we have an HFR conjugation. So again, we have the F factor, we've got an F negative, but here the difference is that we get part of this genetic material ends up inside of the circular chromosome of the recipient bacterium. And so it's that incorporation, DNA recombination. Let's see. So if it's not incorporated, it gets broken down, and the rest that is incorporated gets saved. We talked about nutrition. So if you have photo here, that means that your energy source is light, but you can be a photoautotroph or you can be a photoheterotroph. Photoautotrophs, this is where we're talking about photosynthesis, photosynthetic prokaryotes, and they get their carbon from carbon dioxide. A photoheterotroph does not use carbon dioxide from the air. It has to get its organic compounds from another organism. And that's what makes it a photoheterotroph. So the first part is the energy, where the energy comes from. The second part is where the carbon comes from. We are heterotrophs, and we get our structure from organic compounds. So let's talk about the difference here. So if you're a chemoautotroph, you're getting your carbon from carbon dioxide, but here you're getting it from something like a sulfur compound, a sulfur dioxide, and this is like the deep sea vents. And so you see that in organisms that don't have access to light. Chemoheterotroph is, it has to have another organism for its organic compounds and its carbon source. So we would be classified as chemoheterotrophs because we get both our organic and our energy and our organic from both our energy and our carbon source come from organic compounds. Here are the biofilms we talked about in a little bit more detail. Again, we have it's two or more, and they cooperate cooperate metabolically. So you get the bacteria, you get the slime formation. It doesn't do a very good job, I don't think, of showing the different types of bacteria, but we get then, as it gets to a certain size, we get release. And often these can be antibiotic resistant. So again, they can be really problematic. Another thing we saw bacteria for was nitrogen fixation. And that's where you take the atmospheric nitrogen and it gets incorporated into the soil. And you can look at the steps here as you're reviewing your notes. Then we go into the phylogeny. And this is where we see that we've got our two domains, archaea and bacteria. These are our prokaryotes. And then we have our eukaryotes. So we compared bacteria and archaea a little bit. And I feel like I'm starting to go a little long. So I think I'm going to stop this video here. And I'm going to finish bacteria and do protists in the next video. All right. So that is all in this video. See you in the next one.